One of the most important processes in development is for a cell to divide and increase in number to generate an entire organism. But at some point, these cells must stop dividing, must stop proliferating, and take on some kind of specialised function. And this might be to become a neuron, a nerve cell, or a muscle cell. And this choice between proliferation, so increasing in number, and differentiation, becoming specialised, lies at the very heart of developmental biology. So when geneticists are interested in a process, they look for situations when it has gone wrong. We can then work out why things have gone wrong and ultimately understand more about how the process normally works. What we do is we use a tiny worm called Caenorhabditis elegans and look for mutants. Those are worms where there are mistakes in their DNA, where the control of cell proliferation and differentiation has gone wrong. So I'm going to show you some really cool worms. These worms are wild-type worms, they're normal worms, and they're quite happy wiggling around on the plate. Now, I'm not particularly interested in the whole worm, I'm interested in a specific cell type called seam cells. And these are a bit like stem cells. Now, these normal worms have 16 seam cells. We can see these cells because we tag the gene with a green fluorescent protein. Now, I'm going to show you a type of mutant which is quite interesting. These worms have fewer than 16 seam cells, and that is because there's been a mutation, so the DNA sequence is not correct, in a gene called RUNT1. And RUNT1 is the worm version of a human gene called RUNX. RUNX genes, one of which is found on chromosome 6, are involved in cancers, and they're key in the decision-making process between proliferation, so making more cells, and differentiation, so becoming a specialised cell type. I'm now going to show you a mutant which has too much RUNT1 gene product. So this worm has overexpression of RUNT1. So these worms have too many seam cells, and this is because too much RUNT1 protein is being made. So essentially, I've given these worms a tumour, which isn't very nice. And why these two different types of mutants are interesting to us is because it shows that RUNT1, and therefore RUNX, is very important in the decision to proliferate or differentiate and become specialised. So what we have here is a simple biological system in which to study RUNX genes. And it's hoped that the insights gained from this tiny but really significant worm will help us in the fight against cancer.